Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed, We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
I promised this webinar for maybe like a couple of years to different colleagues. This is a recurrent topic. Many people face the similar projects and they ask like how to proceed. So I was always thinking like it's really better to record it once for many people. So at some point it will be uploaded to YouTube, and I will be able to just reference to the to YouTube video. And of course, I was thinking like maybe I should organize this webinar myself, but then I saw the email from this NGS Competence Network, and I decided that it's a very good opportunity to organize webinar with your help. So thank you very much for this. About the expected level of participants, I start with the basic things for people who probably don't work in the field of the genetics, because I don't want to lose you from the very beginning. And in the end, I will provide some hints for the more advanced people who really face the similar problem here. So basic definitions in the European Union. The definition of rare disease is disease that affects less than 0.05% of general population. Here in America, they have their own definition, I guess in imperial units, I don't know it. There is more than 70% of rare diseases are believed to be genetic. And we will be talking only about genetic rare disease, no prion disease, no other interesting, but not covered in this talk. So each rare disease affects a really small number of people, you can see, but overall, around 3 to 6% of individuals will be affected by rare disease throughout their lifetime, because there are a lot of these rare diseases. So it's each rare disease is kind of, it can look like a small problem, but all of them together, if we sum them up, it looks like a big problem for the society. And I will be my talk will be mainly based on this paper recently published in Nature Medicine several months ago about our approach, how we try to reanalyze like European cohort of rare disease patients. For the details, you can go and read this paper yourself, of course. It will be more in detail than I cover here, because it was an effort of hundreds of people, more than 300 of authors here, and it wouldn't be possible to cover it all in one talk. Yeah, there are hundreds of papers published within this project, and I didn't read most of them, so I just cover my part that was done by me. A bit of terminology from the very beginning. What do we mean when we say solve a case of rare disease? It means we find the combination of genetic variants. It can be one variant, it can be two variants if it's a disease recessive, or it can be some other combination of variants like degenic disease, which is rare, but it exists. And the key point here is like to prove beyond the reasonable doubt that these variants are causing the disease. This is the tricky part. If you don't have this obligation to prove, then you can solve any cases of rare disease. You just put it a more or less random variant, and genome said like, yeah, this gene may play this role because it's included in the pathway. So maybe the patient has this problem because of this variant. That's not how the diagnostic work. We're really responsible, and we really need to provide the high standard to avoid misdiagnosis for the patients, because misdiagnosis it's a huge problem. It can lead to unnecessary treatment, and yeah, so it's a bit similar to what happens in the court. So there is a defense and there is a prosecution, and they try to find arguments pro and contra. That's how we evaluate genetic variants. We choose a variant, and then we try to internally we try to prove that it's causal, and the other part of us tries to prove that it's not causal, basically. So it's a game of probabilities. We're trying to find like convincing probability that this is the cause. Here, what can be another outcome of genetic diagnostics? We can find a variant which partially explains the phenotype, which means the patient may have two diseases. It's not so rare here. So if we find this variant, but we don't explain fully the phenotype, the case still remain unsolved. Important remark, solving a case is a jargonism. Of course, there are no cases, there are patients. It's just like people use this phrase, solve a case. In reality, we should always remember these are real patients with real stories and with real issues. The third outcome is you may find the candidate variant or candidate gene. In this case, you cannot issue diagnostic report for the patient. You must switch to research setting. You need to do scientific work. You need to publish a paper probably with the proof that this variant is exactly what it is. You may do some validation in mice, for example, or like there are a lot of different methods to show, but this is science now. It's not medicine anymore. And the fourth possible outcome is to find nothing with the existing technology. It's not a diagnosis per se if you found nothing. It means that you may try to find the diagnosis later. So you may return and evaluate either existing data or take DNA from the same patient and try to analyze it with different technology. The diagnostic yield, which is like how many cases we actually solve, how many patients find their diagnosis, it varies between 20 and 70%, depending on the standards, depending on the sequencing technology, and the type of rare disease. Some rare diseases are easier to solve. Some, like, I don't know, autistic spectrum disorder, it's very difficult to solve. And like late onset neurodegenerative diseases are also difficult to solve. So it really depends which disease you work. But overall, for now, let's keep in mind like 50%, half of the patients don't find their diagnosis. So they have a disease. It looks very much genetic, but they don't know what happens with them. And sometimes you may even know what disease it is because of the absence of some protein there, but you cannot find the genetic cause. And it's important, even if you know the disease, it's still important to know the genetic cause because it's important for the kids of this patient. So genetic diagnostics, even if you know the disease, it's still important to do this. Uh, like introduction into this probabilistic thing, how do we prove that something is convincing to be causal variant? So this is a bit complex table here. I will explain what's shown here. So let's look at the rows here. Rows show different categories of facts that we can say about the variant, and these facts they will be showing some evidence pro benign or pro pathogenic role of this variant. So it can be population data. If the variant is widespread in population, likely it's not causal of the disease because it wouldn't be so frequent in the population. Segregation data, for example, we have large family and everyone who has the disease, they have this particular variant. Computational predictive data, this is this pathogenicity scores, for example, CAD score, famous things. These alpha missense scores, recent development with the deep neural networks. Then functional data is this functional validation that I talked. So you introduce some genetic change into the cell line and you check how it behaves. And there are like some other categories. And basically you look at the variant and you select the categories. You select this criteria and try to cross some probabilistic threshold. So on top you see odds of pathogenicity. This is like simplification, but it's like numerical representation of how strong this fact is. Is it proving that the variant is benign, or is proving the variant is pathogenic? So you take all these facts about the variant, you sum them up, you multiply the probabilities, and if you cross some threshold of definitely guilty, this variant is causal, then you may write a diagnostic report. If not, then bad luck. Try better. Try to literature search. Try to order some functional assay. You can do better. So this is the it's not averaged, but it's like typical diagram of the journey of a patient, rare disease patient, through the diagnosis to find the solution for their problem. So I will go through this particular example. The patient has first symptoms, then they go to the family doctor, and family doctors are not trained in genetics, not trained in rare disease, and most probably they see this particular rare disease first time in their life. Uh, they can do nothing in this case. Then patient do an online research. Now we have ChatGPT, and it's actually not so bad. ChatGPT is actually, if you put good description what's happening, it can guess quite well. So recently it happened at the workshop in rare disease in München. We were giving a workshop, and one of the participants used ChatGPT very efficiently in this paradigm. So I wouldn't be very skeptical about this. Then after this online research and the visit to family doctor, you get you go to special doctor, and then it often ends up that you don't have diagnosis after this, or the wrong diagnosis, which is the worst. Then you may have a wrong treatment in this case, and sometimes it's even surgery, which is irreversible, of course. Then you go to another specialist, then this another specialist send you to another, and then it's a loop. At some point, you may find the support in the patient organization. It's very important, the psychological support here, and then you finally come up to genetic testing, to realization that it's a rare disease of genetic origin. And as we know that 50% of the patients, they don't have the diagnosis, so you retest it, you do it in some other laboratory, you think that other doctors may find it better. And in the end, if you're lucky, you will get the diagnosis here. So that's a long odyssey which may take years here. So the genetics of rare disease can be really complex. There are many factors which makes it tremendously complex, like the variable phenotype. So one patient with this variant has one phenotype, and another patient with this variant have another phenotype. And like to put them together to show that it's the same cause, it's very difficult. So I'm not going to tell you scary stories about this because I'm not a medical doctor myself. I will tell you different scary stories. I will tell you bioinformatics scary stories, like how it's like bioinformatic bingo. It's the data is incomplete, the data formatted. And when I say the data is incomplete, I don't complain about the data. I think we had amazing data. I think people who put these phenotypic descriptions to the patients, they did amazing job. And it took a lot of time for them to fill all this data for us. It just still, if you have 10,000 of such entries, there still be some inconsistencies, inevitably there. So that's just the property of the data. What was kind of unique in the project I will be talking about, we looked at the data sequenced with 26 different enrichment kits, exome enrichment kits, and it makes this data extremely heterogeneous. So yeah, I will complain about this a bit later. One more thing, in rare diseases, quite fast people realize that we really need to collaborate between each other. Collaboration is a property of all the sciences. It's very important to share the knowledge, but in rare disease, the necessity of collaboration is even of different origin, because rare disease is very rare. Sorry for the tautology, but it's very difficult to recruit a large cohort of the representative cohort of different rare diseases in one sequence, not sequencing, but in one facility. Here in Tübingen, we have a lot of rare disease patients per year, but still, I wouldn't I wouldn't say that we have all the rare diseases in the world. Absolutely not. And to do some studies, to find the causes of these diseases, we need at least several patients there, like to compare. We cannot really diagnose patient, just one single patient here. In rare cases, we can, but most of the time we need some of them to prove that it's actually true what we see. This is just example of knowledge, how the knowledge was developed, and the just example of epilepsies, one particular thing. So what we can see, let's look at the picture. We see on top left corner, this is the evidence that gene is causing epilepsy. And the important fact I want to point you here is the x axis, and there are points. These points are evidence, strengths of evidence that this particular gene is causing epilepsy. So we need to keep in mind this: the genes are not 0, 1. So it's not like we're 100% sure that this causes epilepsy, or we're sure that it's not causing. It's always there is some degree of uncertainty. So when we perform diagnostics, we have to keep even this in mind, not only variant properties, but also gene properties. And on the diagram, it's years on the x-axis. What's interesting here is how the discovery of the new disease genes were happening. I really like this picture. So we can see before sequencing, this channelopathy era, it's people were doing it without genome sequencing. And we see that the one new gene was discovered per several years here. So we develop very slowly. Then sequencing comes, and it looks like the curve goes really up. But what's kind of you see also important, these red points, that some genes were kind of reported that they are causing epilepsy, but they were refuted later. And international collaboration starts somewhere in the middle. So the this ratio of discoveries, it's also because of different facilities, different centers started to work with each other. The problem of collaboration is you work with your patients, you collect the data, you pay for this data from your grants, and then you need to share this data with someone else. And you may be not the first author in the paper in the end. So it's like from the point of the scientific career, it may sound difficult, but for the point of benefits of patients, this scientific collaboration is very important. So in many conferences of rare disease, you will see this thing. People who report, they will say like collaboration is extremely important, and that they put empathies many time there. And I hope I convinced you that rare disease, it's even more than for the other fields of sciences. Collaboration is important here because it's just very rare. And you may find this patient with the same gene affected in, it's actually a real example. It happened with us. We found the patient in Indonesia with the same problem that we have in Germany. I really don't ask ChatGPT to make this picture. I don't know why he put the stethoscope for medical geneticist. But if you ask to draw something medical, it always puts stethoscope here. So just I will be talking about Solve-Rare. Solve-Rare is the big consortium of solving the rare disease. So people decided that within Europe we can merge our efforts together, look at our unsolved patients once again, and try to find the cause there. So this is the map. It's a bit old map. So some scientific centers joined after. The number is showing the. It's not the number of scientific center. It's just a particular facility, particular name here, and the number changed there. Of course, it's a very old picture. But just to give you the idea, it's mostly Western European countries. The big centers of rare disease, they decided that let's do it together. Let's to benefit the patients, we need to merge our efforts, merge our data sets, despite a lot of difficulties from the legal point of view, because sharing the patient data, sharing human data is a very sensitive topic. Still, people decided that let's break these barriers, let's do some additional effort, merge our cohorts, and try to discover something. Try to help to this remaining 50% of undiagnosed patients. These are 4 cohorts that Solve-Rare wanted to recruit in the very beginning. First is just like the existing data that was already sequenced by someone else, like someone in London, someone in Italy, someone in Dijon in France. And other medical centers. So we take the exomes sequenced by different institutes and try to reanalyze them, try to look at the unsolved patients who were not solved before, and try just in the existing data. The second cohort was, let's do some short read whole genomes and long read whole genomes and other we called it novel omics approaches. So we generate the data. Here, third is was the cohort of ultra-rare rare diseases. So even within rare diseases, there are rare diseases. And the final cohort was the unsolvables. And what does it mean? It means that its disease entity, you see the patients with the same phenotype, very similar, but there is no even slightest idea which gene may cause this. So the patients with this disease, which just looks like genetic 100%, they don't undergo genetic testing. The insurance company doesn't pay for this because what's the point to do this if you don't even know the potential cause here? So there was attempt to crack these unsolvable syndromes. That's I will talk a bit about organizational part because it's such a big endeavor. It's very important to look at the organization. It has to work like a clock, the whole mechanism. So let's look at the right part here. What is shown here, like 12 European countries, 37 institutes, and more than 300 collaborators. In the end, it was even more. So this paper in Nature Medicine was describing only less than half of the analysis that we've done. What's also important here, the participants of European reference networks. These are facilities, medical geneticists, like they are unions of different institutions which existed before this consortium. Then the data that we collected, it was more than 9,000 exome sequencing and some short-read genome sequencing for reanalysis. It was generated by someone else. We just downloaded it, uploaded it first, and then downloaded, which is also a huge effort. It was petabytes of data in the end. The information about pedigrees, it's important in the rare disease. So we look at the family structure, who is affected, who is not, because it helps. Then HPO phenotypes is describing HPO terms. This is the example of HPO terms for visual diseases. So on top level, we have more general terms. On the bottom level, we have more specific terms. So in more, the clinician has to describe the patient in some way. So we bioinformaticians would be able to work with this. And this is how we describe phenotype. It's on the tree. So we calculate the distances between patient's phenotype and the genes using these distances on the trees here. So important, interesting thing here. Again, the whole project had to be interdisciplinary, truly interdisciplinary. So on the left part, you see bioinformaticians who participate in the project. On the right part, we see the clinicists and geneticists who were participating, and we had to find the common ground in the end. It was a difficult task because we speak different language in the end, but I think we managed. So bioinformatics were able to work with no, it's not wet lab, it's more like real scientists. Yeah, we were able to work with real scientists. About the bioinformatic part of the project, of course, we had to, the data that we received were done in data freezes. It's just convenient. So clinicians find the cohort of their unsolved patients, and then they submit them. And when we cross some threshold, like in this case, it was like 8,000 exomes, we make a data freeze and process this data freeze first, then second data freeze, third data freeze. And you can see bioinformaticians were working in work groups here, and work groups were grouped by the type of variants. So short variants, for example, then CNVs, the runs of homozygosity. I will be talking today in this talk only about these two work groups. There is no time to cover everything else, and probably there will be no time to cover even these two things. I probably prepared too much slides, but let's see. I promise that we'll be able to have lunch at 12 in 30-something minutes. We don't talk about long reads. This is a paper about long reads published, and I really recommend to read. It's moderately covered, long read 10x, but there is no time. I think important lessons were found in this paper. But we will talk about this reanalysis. We work with exomes today, just exomes. So this is the complex diagram. I will walk you through here. These are the results of the whole project. Of the whole project was described in Nature Medicine, which is less half of the project. We have 6,000 families of unsolved rare disease patients who don't have the diagnosis. The arrow which goes to the right here, it goes to the ad hoc expert review. What does it mean? It means that the institutes which submitted the data to be reanalyzed within this Solve-Rare project, they also perform their own reanalysis. Sometimes it's systematic. So sometimes once per year they stop and say like, let's look at the cases which we were not able to solve 5 years ago, for example. And then they put additional effort. Insurance rarely pays for this, so it's more like reanalysis of the case is more like a scientific initiative, put it like this. So a lot of patients were solved during this reanalysis. So we don't count them as solved within our effort, even though frequently we found the same variant and we say like this is probably the reason. And the answer from these data submitters is like, we already know. We already performed this. We already perform additional analysis and we discovered this. How many patients were able to solve? We were able to solve around 500 of them of 6,000, which is a good result in my opinion. You can see this on the pie chart on the B panel B. The gray ones are still genetically undiagnosed, and the different colors show how the others were solved in the end. So most of the patients were solved with the short variants, which is not the topic of today's talk. But a lot of them were solved just with automated annotated with ClinVar. So just ClinVar is a huge database of pathogenicity of variants, and it's got updated every day probably. Different centers who were able to prove that this variant is causal in some patient, they submit it to this big database. So with the increase of database, the diagnostic becomes easier and easier, I would say. So more than 160 patients were solved just with comparison with the database, simply. 60 were solved with the new genes. So when the patient was analyzed, it was not known that this gene plays the role in this disease. And around 200 were solved via expert consensus. Yeah, it's just re-evaluation by the other people of the same variants. They may find different evidence here, so it's useful. But we'll be talking about the second part, which is looks a bit more a bit less impressive. We solved only 90, around 90 patients with the other variant types, which are CNVs, structural variants, and mobile element insertions. So we talk about these three things. Around 300 patients have candidate genetic diagnosis, which means they are in this scientific research stage. So doctors try to, and the scientists try to prove with these scientific methods that these variants are causal. What are the long variants? Finally, we came to the topic. Introduction is over. What are the long variants and why it's difficult to detect them? Long variants in the short reads normally defined as something longer than 50 base pairs. Why it is longer than 50 base pairs? Because of the tools that we use. We use short variant callers and the long variant callers, and short variant callers start to have problems with variants longer than 50 base pairs because it already takes quite a lot of the read, and it's difficult to map the remaining read on the reference genome here. So for imagine like nanopore sequencing of 5 kb reads, for example, they will also have the issues. They will also have long variants for them, but the definition of long variants for these long reads will be different from this. 50 base pairs is the short read definition of long variant. The problem is with long variants is that they are long. So the rare disease, the problem with rare disease is that they are rare, and with long variants, the problem is that they are long. Here, it's actually true. It's if you cannot read the variant from the beginning to the end, the problem starts. So you don't sequence the whole variant, and then different tricks have to be applied. Structural variants. These long variances are also represented by structural variants. The main type of them is deletion. Simple thing. You just part of the reference genome is removed in the patient. Novel sequence insertion, for example, viral insertion can be. Mobile element insertion is a bit different. Mobile element insertion because this mobile elements human genome consists like by the 30%, probably 27% at least with these transposons who are jumping in the genome. So huge percent of human genome consists of these transposons. Duplication can be tandem, can be interspersed, and it's very important to distinguish between them because the consequence of tandem duplication is different from the if interspersed duplication, if the duplication happened in some known disease-causing gene and it broke the gene, then it's important. But if it's some interspersed duplication somewhere in non-coding in the genetic desert, you don't care about this. Yeah, there are also inversions, translocations here, and these are the how you detect them with the short reads on the left. Like Illumina made this trick with the paired ends, paired end reads. And look in the orientation of the reads and the insert distance, you can judge if something structural happened in the genome here. In exomes, since we sequence only a small percentage of human genome, we don't sequence breakpoints of the structural variants most of the time, and we can rely mostly on read depth. So most of the people, they do only read depth analysis, and you can it's the second column here, and you can see how many variants we cannot detect. We cannot detect novel insertions, inversions, like anything copy number neutral. We can detect only copy number changing events, and we cannot distinguish between tandem duplication and interspersed duplication. The last two columns, the split reads assembly, they're of course they're very important for long read sequencing, but in short read sequencing, they're more limited success. We'll be looking at the detection with read pairs, orientation and distance, and read depth here. These are three types. It's called beyond standard analysis that patients were able to solve with our bioinformatic analysis, which were not solved before with the other bioinformatic analysis, which was done before, because all these patients were kind of institutes were trying to solve them by themselves. First, panel A is the de novo copy number change, copy number variant. It's deletion here. Second is the mobile element insertion, and third is the structural variant. So we went a bit beyond, and we tried to detect mobile elements in exomes and the structural variants in exomes, which people don't normally do because the outcome is very limited here. CNVs, you may say that people detect CNVs in exomes. It's a routine thing, but CNV detection is difficult. It's very noisy, the data, and some expertise has to be bioinformaticians has to have this expertise on top of the standard to be able to successfully perform CNV detection in exomes here. Okay, so let's run through these variants, three types of variants: mobile element insertions, structural variants, and CNVs. Here, first is mobile element insertions. It was a paper led by Robin Wingard from Nijmegen. It was amazing paper, not only diagnostic, but also bioinformatic benchmark of the tools that were used. What are the active transposons? There are three types of active transposons in the human genome. L1, big, huge one, 6 kilobases, SVA, 3 kilobases, and the tiny one is ALU, but they're most frequent reason for the medically relevant mobile element insertion is this ALU insertion. What is here? Within this study, we performed the benchmarking of the tools, and it was very difficult to to make a gold standard of variants for exomes in exomes. So it was solved in quite elegant way, but with a lot of manual work. And if you want to learn more, you can read the paper, and we can see that in exomes, two tools perform the best: is melt and scramble. So I recommend if you perform the such reanalysis in the future, you use melt and scramble tools because they have the best performance among them, and also their melt is not very fast, but scramble is quite fast in my opinion. So I share a bit of details for people who are advanced and who want to maybe use it. First of all, it's very easy to estimate allele frequency of these variants because the resolution is typically one base pair, and you can do it really easily, like with short variants. And the second is it's important to distinguish between the insertion of mobile element and the mobile element mediated structural variant. So we know that 27% of human genome is consisting of these old mobile element insertions, which happened in the past in the, I don't know, at the stage with humans were apes maybe. And if genome breaks there, your caller may say that this is mobile element insertion, but it may be not. So if you write a paper, for the patient, it doesn't really matter what broke his gene. what caused the issue. But if you write a paper, it's important to distinguish between these two. The first example here is the patient who had family who had a benign familial chorea, and you see four IGV tracks on the panel C here. Panel B is the MRI showing the medical side of the question, but we care about bioinformatics. So this is the panel C we look for, and we can see at the two bottom tracks we see these rainbow stripes there. This is a typical signature of a mobile element. And the red stripe on the right is this poly A tail, which is the thing that you see in mobile element insertion. The lesson out of here, the whole exome, when we performed the generalized QC based on coverage, this sample was not covered enough. So this sample should be in theory filtered out by QC control. But if we care not about our power of detection of the variants, but about the solving existing exomes, then we don't really care about this coverage. So when you perform QC control, be careful, don't overperform it. For mobile insertion, it was enough. This 30x coverage, it was enough to detect the reason. This is another example. It's insertion of in one gene. Another lesson from here. You can see a gene structure on the bottom on this second panel, and you see that the mobile insertion happened quite far from the exon. This is non-coding thing, but it's still causing the disease. Bioinformatically, we can perform automatic matching between patient and the expected gene disease phenotype. It's important to apply this tool. So you would miss it if you perform standard filtering of non-coding variants. Here, second topic is the copy number changes, how we applied, how we detected copy number changes in these 6,000 unsolved families. First of all, heterogeneity of the data. This is the coverage of exome enrichment kits. So each on the x-axis, each of these violins is the different exome enrichment kits. So we can see old kits, for example, Agilent Sure Select version 1. It was released, I don't know, 20 years ago or something like this. And the coverage was limited back in times. And normally it increases with the more newer kits. So Agilent version 1, 2, 3, 4, 5, 6, you see, and it kind of increases up because sequencing became cheaper here. But it's kind of general observation, but some of the kits have quite lower coverage and they're new. The Twist kit and the IDTX kit here. Why so? Because the producers of the kits, they managed to make a good evenness of coverage. So these new kits, they have a different nature. So it's easier to detect copy number changes in them despite the moderate coverage. So you don't need to sequence them to 100 something X anymore. The general rule, like better, bigger, the coverage, easier the detection, does not work here. In exomes, it's all about signal to noise ratio. How well is hybridization of probes in your kit works. Here, so we use three different approaches because Barcelona, Nijmegen, and the Tübingen were doing three different analyses. First was the clean CNV. It was my PhD tool. So yeah, it works. I described the principles, how it works. So different bioinformatic algorithms were used here. Yeah, it used what's important about clean CNV approach that exons were divided into windows. So we were not working with the producer provided target definition, but we divided into 120 base pairs windows. Then exome depth tool, it's a good tool actually. And it uses hidden Markov model and quite advanced, robust beta binomial modeling, despite it was developed 13 years ago, something like this. And the third tool is based on SVD. This is this matrix transformation, let's call it like this. So we remove the noise from the data using these PCA-like matrix operations here. There are pro and contra here, of course. When we perform this SVD, and it's written in the paper, we can miss aneuploidies, like chromosome X triplication. You can miss it because it's just too huge, and it will already affect your SVD decomposition. So you have to be really careful with these things. And many people who use these tools, they don't read the paper, and they don't know this, and they expect to be able to detect large variants, but they don't. You will see it a bit later. So it's very difficult to estimate the allele frequency here. And people, most of the time, they ignore multi-allelic and polymorphic CNVs in the human genome. You cannot detect them with the standard CNV color. You need to use the catalog, which someone generated with specific methods, just because the assumption of all CNV colors is that the data is deployed on baseline. The majority of the cohort is deployed. It's not true in the case of multi-allelic CNVs. Here, so the QC control, yeah, it was very important. So we had two choices: perform uniform QC control, and then probably would select only the best kits, and then many groups will receive no results from us. It would be quite bad. So we decided to be less stringent, and every group submitted with maybe old kits or noisy kits, enrichment kit, they still get some results from us. This is the table of how many variants were generated and survived the filtering that we had. Why I show these numbers? Each of these number is actually entry in Excel table, and the clinician has to go through all of them. So these hundreds things, it's something that a person manually went through. So the numbers are huge. Numbers are impressive. So this is the diagram of length of detected CNVs. What would you expect? Short variants should be prevalent here. So you should detect more short variants than the long variants, because long variants are more damaging and they are eliminated from the population, of course. Even in the rare disease cohorts, 99.99 calls, CNV calls, will be benign here. So this is, it can be used a bit of like a quality control. These are the results. The blue ones are duplications, red ones are deletions. The top panel shows the disease causing, so it was proven that this variant is disease causing. B shows the partially explaining phenotype. So, for example, triploidy of chromosome X. It can often explain some of these symptoms. And the third is the candidate variants. So these are performance of three tools. So 3 lines are 3 different tools. And the ClinCNV, the tool that I developed, performed the best in terms of detection of short variants here, because it's not because the modeling was advanced, because I applied some trick. I will talk about it a bit later. Here, this is the trick. So on top, we see the producer designed the probes, exome enrichment probes, for these intervals on top. What we do, we divide them in 120 base pairs windows. Why 120 base pairs? Because the probes, hybridization probes used by Agilent, were exactly 120 base pairs. So this is the optimal window. And there was a paper published, not by us. They tried different windows and they found out that 120 base pair magically were the best window size. For normalization, it was the best. Yeah, no surprise. It's because how it was designed. And the third one, it's quite complex. If you just use these overlapping windows and you try to call CNVs in them, your data will be very much correlated between each other, because the coverage, it's affecting the neighbors here. So it's the last step, the third line, the third track in IGV here. It's how we subdivide everything, and then we try to divide coverage in a way so it loses the correlation structure here. This is how it looks. In the end, this is one of the samples, and this is how the coverage looks. The red one is indicating deletion, and the blue one indicating duplication if it's far enough from the baseline here. And this is how it looks for the interpreter. So we generated screenshots for each variant, and we use the best existing artificial intelligence, the best existing neural network nowadays, humans. So if you're trained, you can look at this complex plot and you can immediately say if it's false positive or if it deserves the second notation or not. You just scan it from the top to the bottom without these convolutions or something. Humans can do this after some training. It looks complex here, but that's how we operate it. It's just the results of different colors plus some other informative tracks. We don't have time to go through the details here, but it's instead of going through many Excel lines, always visualize the variants. It would be for long variants, it's the way to go. This is one of the examples that we're able to solve a large family of Mexican origin. They had the colorectal cancer, and the first time they went for genetic testing, it was 2003. So it took 20 years for them to obtain the diagnosis. They had the colorectal, diffuse gastric cancer, sorry. They had them in really age, like you see, this is the family tree, diagnosed at 27 years old, diagnosed at 28 years old. So very young, 23 years old. It was, yeah, it sounds quite tragic. 20 years took from the first genetic analysis for us to find the diagnosis. And by miracle, we were able to recontact this patient. They moved back to Mexico back in then, and most of them already did the prophylactic gastrectomy. Yeah, so they already performed surgeries because I didn't know who of them are affected, who are not in this time. So we found a solution after 20 years, but it was probably a bit too late. Last part of the talk, of the informative, last informative part of the talk is structural variants. Again, I remind you, we look at the orientation and distance, insert distance between reads to detect them. Details of analysis we used only Manta. We do it very simple. The breakpoints have confidence intervals, so evaluating of allele frequency is not easy. And it's useful to determine not the whole structural variant allele frequency, but each breakpoint allele frequency, because of course, some of the real disease-causing structural variants can have one breakpoint in the frequent place where like genome breaks frequently, and other in the place where the genome doesn't break frequently. Yeah. These are the results that we have. We divide in three categories: simple CNVs, complex events, and the copper number neutral. Everything is in exomes. It happens in exomes. So we detected two inversions in exomes just by chance here. And what's important here is the was it detected in parallel with CNV analysis? So we're able to find 8 structural variants which were not detected by CNV analysis here. These are the examples here. So on the bottom, you see two inversions, how they look in exomes here in IGV. Second is the complex event. We see duplication and deletion next to each other. And the first one is really short duplication, but it was enough. It was like 80 base pairs, but it was enough for JATK to miss it because it's already long enough. It's longer than, it's still shorter than the read, but it's already impossible to detect them. Last things like we more or less finished with science. Let's talk about a bit the educational part. So this is the important concept that we came up during working on Solve-Rare. It's how to make bioinformaticians and the clinicians speak the same language. This is the concept of solvatons. There are 4 of them. It's like a hackathon, but we try to solve the patients instead of develop the software here. So the ATF, which is Data Interpretation Task Force, they provide patients, Data Analysis Task Force, the bioinformaticians. They perform some analysis, and then we meet all together in some location. And instead of doing the just conference or workshop, we just go through the patients, which were like clinicians are going through their own patients, through their results that generated by us. And we are able to, since we are there, we're able to show the hidden artifacts of bioinformatic tools there. So they have the opportunity to come to us and ask like, I see this in the genome browser. Does it make sense? And since we are bioinformaticians, we know how algorithms works, and we can say something is wrong, for example. Here, so that's 4 of them. First was about RNA sequencing. Second was about long reads and optical genome mapping. Then short read genomes, and the multi-omics, which included methylation, RBS. It was the reduced representation sequencing, and the RNA-seq also. This is the numbers of participants from different countries. The green shows the clinicians, and the blue shows the bioinformaticians here. So these are like how many participants were able to present on site, how many were online. One more thing, it's a similar event, undiagnosed hackathon. It happened in Karolinska in 2 years ago, and 1 year ago it was in Nijmegen. Why I show this? These are a bit different events here, and in the Solvathon you don't have chances to get in because it's internal thing, and you really have permission to look at the data here, and it's difficult from the regulatory point of view. But this undiagnosed hackathon, if you really want to participate in, if you really need to, you have a talent of solving cases, it makes solving case, I mean, finding diagnosis for the patients. It makes sense for you to apply for one of these events and join. So these are people with absolutely different specializations here. Clinicians of absolutely different profiles, and even patients were here because sometimes it's important for many different clinicians to examine the same patient to look at the things that maybe was missed by the one particular specialist. Multidisciplinary board is important. It's just several days, so we try to solve the case, find diagnosis for the patients in the several days. It's an amazing event. There is a successor of Solve-Rare more or less, a successor of many different consortia. It's called Ardera, and we started just half a year ago. And you see, it's a big European Union presentation, and the even non-European Union. For example, there are many institutes from Turkey, from Georgia. So I encourage, if your institution is ready to wants to join and do this reanalysis together, it's a possibility. So you see different huge consortia in the end merge their expertise to form this project. What's unique about this, that we want to move from the distributed analysis to federated analysis. So instead of collecting the BAM files and the FASTQ files from the different institutes in one data center, we will try to, in the end, come up to the idea how to do bioinformatic analysis separately. So we do bioinformatics here, with other do bioinformatics in Paris, for example, and then we merge our results together and able to find new insight about rare disease without the actual sharing of data, because sharing of data is complicated from a legal point of view. So that's more or less it. I encourage everyone to who leads the group in this to contact responsible people. And thank you very much.